across the tree of life, whereas others are vanishingly rare. And I'm going to use flowers as an example. So Stebbins was one of the first people to ask and address this question in a paper published in 1951. And he looked across all families of angiosperms and classified them as being characterized by what he referred to as the primitive state of one of these 8 characters, or the advanced state of these 8 characters. And I should say that he chose these 8 because they are often used in classifying angiosperms. So he scored all the families 0 or 1 for these 8 characters. And if you think about 8 binary characters, there's 256 different combinations of these 8 characters, and these are represented by the 256 boxes on this chart. The numbers inside the chart shows the number of families that are characterized by that particular combination. And you can see that definitely the character combinations are not distributed uniformly across this space. There's a heck of a lot of white space here. There are a lot of cells that have very few families, but there are also cells that have a lot of families. And so Stebbins looked at the distribution of these data, and he wrote, families are clumped in trait space, therefore combinations of characteristics found in the larger, more successful families of flowering plants evolved through the guidance of natural selection because they represented particularly successful methods of solving problems of fertilization, seed dispersal, or both. So he looked at these combinations of characters that were highly distributed and argued that they were very common because they are adaptive. But there's another possible explanation that these particular combinations of characters could be common because lineages that possess them diversify at a greater rate or go extinct at a much lower rate. And which brings us then to the questions, the specific questions that our group wanted to address. What explains this non-random distribution of character state combinations? Is it natural selection for particular combinations or differential diversification of lineages with those combinations? Which characters actually matter to this uneven distribution? And do character combinations matter? So in order to get at these questions, we needed a way to distinguish between natural selection and diversification. And we needed a way to look at multiple characters simultaneously. And our approach was to use SSE, or state-dependent speciation and extinction methods. And I'll point out that Brian Amara and Jeremy Bellew just published a paper about not throwing the baby out with the bathwater. So we used this kind of approach. We used a stratified random sampling of the angiosperms, and random sampling is required for these SSE methods. Sampling species, or from species from GenBank, sampling sequences of 7 loci that we used to infer phylogeny using a family-level constraint tree, because we weren't really interested in reanalyzing the phylogeny, but we needed branch lengths for the analyses, using 40 fossil calibration points, scoring characters, and then using an extension of existing SSE methods that I'll tell you about in a minute. So what characters? Well, we went back to our inspiration, this 1951 Stebbins paper, and looked at the 8 characters that he used, and chose 6 of those because these characters influence plant-animal interactions, and we thought they might be subject to strong selection or influence diversification. And we coded all of the taxa that we sampled from GenBank as to whether they possess the inferred ancestral or derived state of each of these 6 characters. So if we have 6 characters, each with 2 states, we have 64 possible combinations of those characters. So here I show each of those possible, each of our 6 characters with the 2 states, the inferred ancestral or the inferred derived, and each of the columns represents one of the 64 possible combinations of these characters. And so how was this distributed in our data set? So here I show the actual frequencies of the character combinations and the frequencies expected if those character combinations are distributed at random. And so you can see that this is clearly a non-random distribution of those characters. In fact, it's highly skewed, with over 50% of taxa here having the 5 most common combinations of characters, and over 50% of character combinations not represented in the data set at all. Now remember, Stebbins looked at the same kind of data and said, well, these are common because they're adaptive. So now how are we going to get at that question using SSE approaches? Well, we can think about a character in state 0, and every time there's a mutation in that character with the origin of the character state 1, the distribution or the balance of those two character states is going to be determined by evolutionary processes such as natural selection, drift, and reverse mutations. But we're not interested in only 2 characters, we're interested in multiple characters, so we can add in, now we have 2 bivariate characters, 4 possible combinations. Again, we can, these evolutionary processes will determine the distribution of those character states. So we have this sort of transition taken care of, but now we want to explore the influence of diversification. And so we can add in speciation rates, these blue areas. So when lineages that possess that particular combination of characters speciate, it increases the frequency of that character combination, and extinction will decrease the combination of that character. And using these SSE approaches, we can calculate these rates. So for example, in this 2 bivariate character combination example, we can calculate 8 transition rates, 4 speciation rates, and 4 extinction rates that would determine the distribution of this character combination, these character combinations. But of course, we're not interested in 2 characters. We're interested in many, many characters, or at least in our data set, we were interested in 6. And so we have now 384 transition, 64 speciation, and 64 extinction rates to calculate. And as enthusiastic as we are about character correlations, we can't estimate the full model. So our solution then was to create what we call focal areas. And so we partitioned these data into what we call the focal area, which is all character combinations that possess a particular character combination. So in red I have here all the character states that include, for example, corolla present, bilateral symmetry, and fused stamens, regardless of the state of the other 3 characters. So those comprise the focal area, and everybody else gets called non-focal. And you can sort of pull that out to see that now we have sort of 2 areas in this complicated trait space. And now we can calculate 4 transitions, 2 speciation, and 2 extinction rates using this focal area approach. But it's not just one bipartition, so we used all possible ways that you can bipartition this data set into focal and non-focal areas. Okay, so in all we had 728 focal areas, 5 transition models, and 6 diversification models that we considered, for a total of 19,657 models that were evaluated. Likelihoods were estimated using diversity, and AIC and Akaike weights were evaluated. And when I say we, that's Brian O'Mara and Stacey Smith, who figured out how to do this approach that they named the famous approach for focal area, multi-state speciation and extinction rates. And these 2 are by far the most brilliant scientists that I have ever had the pleasure of working with. They're just fabulous. So what did we find out? What time is it? What did we find out? Well, amazingly, 99% of all the AIC weight was concentrated in just 10 of these 19,657 models. And all of those models identify bilateral symmetry as important. But amazingly, so the best fitting model identifies corolla present, bilateral symmetry, and stamens few as the critical combination of characters. And these 3 characters were present in the majority of these top 10 models. So character combinations, right, combinations of functional characters are clearly important in this analysis. Okay, so what about diversification and transition in determining the distribution of character combinations? So starting with diversification, so I've gotten rid of the weights and stuff, and so here's just the focal character combinations. And here are the diversification rates. This is the diversification rate in the focal area, the diversification rate in the non-focal area. And you can see for the top model, diversification of lineages that have this focal combination of characters is nearly twice the diversification of lineages that have any other combination of characters. And in 8 of the other 9 top models, diversification rates within the focal combination are much greater than diversification rates within the non-focal area. So diversification rates are clearly critical in affecting the distribution of character combinations across our data set. So what about transitions? So here I show the transitions, and it's easiest just to look at this column, showing whether the transition rates that were estimated for within and among these different areas are equal or free. And you can see that in the top model, all of the transition rates are equal. So there's no evidence for biased transitions among any of those character states. And in fact, models with differential transition rates had less than 30% of the AIC weight across all of our 19,657 models. So transitions, differential transitions, are not important in determining the distribution of character combinations across our data. So going back to the question that I started out with, what explains the uneven distribution of character combinations across the angiosperms? Well, we find no evidence for differential transitions, and by inference, no evidence that strong selection for particular character combinations plays a role in determining the distribution of character combinations. In contrast, differential diversification rates are strongly supported. Greater diversification is associated with 3 key characters. You made me move my buttons. With 3 key characters: corolla present, bilateral symmetry, and few stamens. And we argue that these 3 character states relate to the precision of pollen placement and perhaps facilitate speciation. And so let me back up and explain this a little. So you'll notice that corolla present is part of these 3, right? And which suggests that bilateral symmetry of the corolla is much more important than when bilateral symmetry is contributed by any other of the flower characters. And if we think about bilateral symmetry and stamens, bilateral symmetry really controls how pollinators enter the flower, constrains its position with relation to the stamens, and then few stamens allow very precise placement of that pollen on the body of the pollinator and then pick up that pollen, and so may facilitate prezygotic or pollinator specialization and prezygotic isolation. And so these characters may facilitate speciation, and critically, those characters need to function together. Okay, so 2 minutes and I have an answer, but I'm not done yet, because if corolla present, bilateral symmetry, and few stamens, if that character combination, lineages with that character combination diversify faster than those with other combinations, then why isn't that combination the most frequent? We actually have only 30% of the species in our data set are characterized by this combination of characters. So we actually used our diversification and transition rates to estimate the expected frequencies at equilibrium, and we calculate that our focal combination of characters should be represented by 90% of the angiosperms at equilibrium, but we only have 38%. We also calculate that most of the angiosperms, you shouldn't be able to find this root state anymore, but we still have about 10% of our data, our taxa are still in that root state. So what gives, right? And so we thought about this, and this is multiple characters, right? And so you need to assemble this combination of characters before you kick off this diversification rate. But transition rates are really slow, right? So it takes a long time to get those characters to come together. And so we explored this a little bit further with some stochastic modeling. So here we estimated, again, using these traits, or these rates that we estimated, what's the first instance of the appearance of that character away from the root state. And these distributions here show all the different first occurrences in our models with the big bar, the median. And so what you can see here is that it takes about 11 million years for the first transition to occur in anywhere, and it takes about 71.5 million years for the very first occurrence of our key combination of characters. So there's very significant delay, and I would argue it's imposed by this really low transition rate, before we assemble the characters necessary to stimulate the diversification, the kind of diversification that's going to drive character distributions in the angiosperms. When we look out across the angiosperms, the distribution of character combinations is likely far from equilibrium. So in conclusion then, the character combination of bilateral corolla symmetry and few stamens is important for floral diversification. This combination of characters relates to precision of pollen placement and speciation. Differential diversification rates are more important than differential transition rates in explaining character combinations, but transition rates are the limiting step. They're the rate limiting step when we think about evolution over the long span, and these transitions delay the assembly of key character combinations, meaning that distributions of character combinations are likely far from equilibrium. And to the extent that most of the characters that matter to organismic function require multiple characters, then the importance of the transition rates that assemble these multiple characters then are going to be critical in the way that we understand the pattern of diversification over the macroevolution timescale. So, thank you.